So in the practical about osmosis, right, apart from using whisking tubing and using agar, okay, whisking tubing or using agar cubes, okay. So what are other things that you can use in the practical to illustrate osmosis is, of course, plant cells lah. Okay, plant cells. So for plant cells, you know that whether there's a net movement of water into the cell or out of the cell actually depends on water potential of the cells versus water potential of the environment, right? So they may ask you to do an experiment to find out what is the water potential of this plant cell. So how are you going to find out what is the water potential of the plant cell? And then the plant cell that they can give you, okay, usually what do they give you? They can give you, why is it become blue color? Okay, so they can give you, let's say, for example, onion epidermal cell. Hang on, let me type here. Okay, onion epidermal cell. So usually onion epidermal cells, you know, you take the onion, you chop it off. So you take the onion, take the onion. This is onion, no? Okay, onion. Looks like a moth. Okay. And then you chop it off, right? Then you can see that you got scale, right? The onion scale, like, you know, fish scale, right? So to the inner part of the scale, you peel out the skin. Peel out the skin of this inner layer. So that skin is a tissue. That tissue is called epidermis. So it's called onion epidermis. So we want to observe osmosis process in those cells, right? Like those micrographs I've shown you before. Cell undergoing plasmolysis, cell that's turgid, and so on. So for that kind of osmosis, we use onion epidermal cell. Okay? And yeah, all right. Also, we can also use onion epidermal cell to find out what is the solution that when the onion epidermal cell placed in, give the same water potential as the cell. That means we actually want to measure what is the water potential of the cell. Be it onion epidermal cell, be it sometimes they give you potato, okay? Sometimes they give you celery, okay, celery. So whatever plant cells they give you, they may ask you to find out, so what is the water potential of the cell? So the basic principle is if the water potential of surrounding equals to water potential of the cell, right? Psi of the cell, okay, we start with cell first, cell is more important. Psi of cell is equals to psi of environment. That means I will expect what kind of net movement of water? Charmaine? Net water movement.
Sorry, teacher, can you repeat?
So when the psi of the cell is equals to psi of environment, what is my net water movement?
0.
0, no net water movement, right? So this one you learn already, isn't it? 0, 0, 0. Okay. So when psi of cell, so this is hang on. I called Charmaine just now, right? No thanks. Give me Charmaine. Where is Charmaine? Charmaine. Oh, S is actually S. 2 points, 2 points, okay. So you all know what are the rest I want to ask already, right? So when psi of the cell is more than psi of environment, what will be the net water movement? Januli, I call Januli, I call somebody else. Li Pei Yi.
It will move out of the cell.
Correct. Yes, region of higher to lower, isn't it? So move out the cell, correct. So you move out the cell. So let's put that net water movement is out. Okay, so this is out. Then if the size of the cell is, wait, just now I call who? The Pei. Pei, or these 2 points very easy to get. Congratulations. Okay, so if the size of the cell is less than the size of environment, what will happen to net water movement? Ong Shu Min?
Into the cell.
Net movement into the cell, right? So this one all of you know, isn't it? So now for the environment, I'm using sucrose solution, right? Molarity means different concentration of, in the form of molar, okay? Mole per dm cube, molar, okay? So therefore, when I want to find out what is the water potential of my plant cell, I need to find out what is the concentration of sucrose that when I immerse my plant cell into it, there will be no change to my plant cell. That means because there's no net water movement. Okay, so if you look at the y-axis, they are using strips of daffodil corona. Daffodil corona is one of the part of the flower, okay, of the daffodil flower. So this is initial length versus final length. So usually when they give you potato, for example, they will be giving you potato in the form of potato chips. No chips. Potato fries, yeah, okay. So it's like the shape of potato fries, okay? So either that means you're getting potato like this, okay, like potato fries up, but then the measurement is all very accurately measured one, okay, length, height, and so on. Or they can give you potato rods, okay? So the lab assistant will have actually made potato rod out of your potato tuber, right? You have to cut it out, right, into this, okay? Or celery also very long, so you have to cut it into maybe segment of don't know how many cm length, right? Celery also can. So when you place all this potato rod, okay, or cube or the celery into the sucrose solution of different concentration, you need to allow time for osmosis to happen. So usually for osmosis, you need at least, I think for me, for potato rod, 30 minutes, okay? Minimum also 20 minutes in exam lah, okay? Minimum 20 minutes to allow water to move until equilibrium is reached. Usually 20 minutes equilibrium cannot be reached lah. It must be more than 30 minutes, but 20 minutes you can see the result already, okay? So then you calculate initial length versus final length. So that means this is your potato rod, right? You put it into 5 different concentrations of sucrose. That means you got a beaker. It's not 5. You see here got how many? 1, 2, 3, 4, 5, 6. So actually, in this experiment, there are 6 different concentrations of sucrose that you're going to use. How come they don't use 0? They use 0 also can, right? 0 is also one of the concentration. Okay. So I will use 0. So therefore, I have got 7 already. I got 7 beakers, including 0. Oh, they didn't use 0. They use 0.1. Okay. 0.1 is the last one here. Okay, so for every beaker, I got a fixed volume of sucrose solution. Remember, everything must be the same except for one thing, which is the sucrose concentration. So you got 7 different sucrose concentration. So the volume must be the same. Then your potato rod must also be the same length and same number. Let's say if I put 1 rod inside, that means all of them must be 1 rod of the same length, same diameter. Everything must be the same, except for the sucrose concentration. So I leave them, all of them, I time them the same time for 20 minutes to allow osmosis to happen. After that, I take out the rod and I measure again. Then I calculate initial length versus final length. So after I calculate, I know that If the initial length and final length is the same, that means I know that there is no net water movement. That's why it's still the same. That means for that concentration of the sucrose, the water potential of the sucrose solution will be same as water potential of the cell. So that's how I can know the water potential of cell. That means from here, I must interpolate to my graph that I plotted from the result of experiment. And then from here, I find out. So what is the concentration of the sucrose that gives no change to my potato rod length? So this is the concentration, but this is concentration. I want to convert concentration okay, which is in molar, to psi, which is water potential of the cell. Okay, because I want to know water potential cell and water potential cell in, let's say, kilopascal. So how am I going to convert that after I found out the sucrose concentration? There is a table, okay, that is available for you to find out from the table. If this is the sucrose concentration, what is the water potential of this sucrose concentration? So therefore, you can use this table to find out. Okay, so this is my sucrose concentration. Okay, and it is in molar. And then this is my water potential psi. Where's my psi? Water potential psi. And this is in kilopascal. Or atmosphere also can. Sometimes the unit is in atmosphere or millimeter mercury. Okay, so kilopascal. So from there, I go to this table. I look for the value here. So this value is how much? 0.45, right? So I look for the value, which is 0.45 here. And then I read. So what is this value for psi? If my sucrose concentration is 0.45 molar. So what is my value for psi? I read from the table. So with that, I will be able to find out what is the water potential of my potato cell. Okay. So this is one of the easiest way, right? Can we add a little bit of five minutes more to elaborate on this? Now, you see, if those who cannot stay, you can leave, okay? Those who can stay, please stay, because I'm not finished yet here. Now, if I use the rod alone, right, the surface area to volume ratio of the rod is actually very small. If I want a diffusion or osmosis to happen, okay, I need to increase surface area to volume ratio so that the rate of osmosis is faster, so I get a bigger change in a small time for me to see the reading. Okay, so you want to plan your experiment in such a way that use minimum money, minimum time to get maximum result. So therefore, I may want to cut my potato into small discs. So I may cut this potato into, let's say, 10 discs out of this rod. So when I cut into 10 discs, can you see that immediately I increase the surface area to volume ratio of this potato rod already? So therefore, the rate of osmosis will be faster than the original experiment. Okay, but then when your potato disc is 1 mm thick or 2 mm thick, right? It's very difficult to measure the thickness after changes because it's very small. So if you cut it into potato disc, right, this usually we do not measure the change in the thickness of the potato disc. Okay, we will measure something else, and this is very important. That's why I need to mention here. So what do we measure in that case? Okay, so same thing. We prepare all the 7 different concentration of sucrose solutions, and we put all the 10 discs, okay, of the same size for each of the sucrose solution, and leave it there for 20 minutes. After 20 minutes, we allow osmosis to take place, right? Okay, so there'll be one beaker where there's no net movement of water into the potato disc. Let's say this one, no net movement. Okay, I got 10 potato discs here. I draw 2 only. Draw 2 only. Okay, so say for example, in this particular sucrose solution, It's higher concentration. So if it's higher, means is there a net movement into or out of the cell? Xin Yin Ding.
Teacher, can you repeat the question?
In this solution, it's the same concentration. Sucrose concentration, same as the water potential of the cell. Now, this is a higher concentration of sucrose. Is there a net movement into or out of the... Hang on. Hang on. Go back to Xin Yin Ding. Net movement into...
Out of the cell.
Out of the cell. Yes, correct. So out of the cell, because more concentrated, right? So therefore, the water potential in the cell is higher than here, right? So there's net movement out of the cell. Okay, net movement out of the cell. So when you allow the water to move out for 20 minutes, what will happen to the concentration of this sucrose solution? Will it increase or decrease or remain the same? Who I've not called today? Who I've not called today? Nobody. Philbert.
Concentration decrease.
Concentration, correct, because water, right, from the potato cell go into the sucrose. So sucrose is being diluted, right? So the concentration will decrease. Okay, very good. So how will I know whether there's movement of water out of the cell or no movement at all, or movement of water into the cell? I cannot measure the disk because the disk is too thin. I need to measure the water. Sorry, I need to measure the sucrose solution. Is there an increase or decrease in concentration of sucrose? Can be measured like this. So I'm going to use blue color to show you. Now, I'm going to use a boiling tube. And in this boiling tube, I'm going to fill it up with the concentration of sucrose, the initial concentration I use in this tube, in this beaker. This is the initial concentration, okay? So after 20 minutes, I will take a pipette and transfer a small volume of this solution into here. So basically, I got a pipette, I put it into the middle of my boiling tube, and I will squeeze, okay, 1 drop of this solution that I obtained from here after the experiment into this solution, which is the original concentration of sucrose of this one before experiment. So we say that water enters the sucrose solution, right? Sucrose becomes more diluted. What do you think will happen to this drop of sucrose here? Will it remain suspended, or will it float, or will it sink? Okay, suspended means they remain in the same level, suspended, okay, or floats, or sink. Okay, who wants to try? Who I've not called yet? Shannon, I call you already. Shannon? Shannon?
I'm not sure.
Okay, open to the floor. Type your answer. First one correct, get the marks. This drop is more diluted than the original solution. Okay. Okay, you see, whenever you want to consider something in the water will float or will sink, you always consider density, right? Correct or not? Anything that's less dense than the liquid will rise. Anything that's more dense will sink, right? And what is the formula of density? Hoshini. I cannot call it Hoshini because Hoshini got mark. Can. Okay, Hoshini. I've not called you yet.
Mass or volume.
Mass or volume, right? Okay, very good. So density is mass over volume. Mass over volume, is it concentration? Kirti, is mass over volume concentration? Yes, isn't it? Okay, how do I add marks for okay, +2. Kirti, yes, okay, correct. So you only, now you get yourself 2 points. Because concentration is actually number of moles, isn't it? Per volume is concentration, right? And mole per volume is proportional to mass per volume, because mole is proportional to mass. Also, mass is proportional to mole, right? So therefore, this is actually concentration. So when the concentration of this solution has decreased because diluted already. So when the concentration has decreased, that means the density has decreased. So the density have decreased, that means when you release this solution into the original concentration, because it's more diluted, it's less dense means it will float. Can you see that? So the moment you release it, if it floats, you know that, oh, okay, so water has moved out from the potato disc. So you have to test all of them and see which one the water droplet remains suspended. So that is the one that the concentration of sucrose will be equals to the water potential of the cell. So this is another way to find out which concentration of sucrose tally with the water potential of the plant cell. Okay, this was an experiment that was given in exam 2 times. 2 times in 2 different years. Okay, this particular question. And also they've given agar blocks for osmosis. They have given celery for osmosis as well. Okay. Potato rod also they've given it for osmosis question. Right. One more I've not taught you. Are you okay to continue? Last one. That we will complete the whole session already for osmosis. Because that one I've not taught the previous class. Can or not? If you need to leave, you can leave, okay? Because I want to finish everything. Okay, now the last one is this. You can actually count the percentage of plasmolysed cells in the microscope in the sucrose solution to find out at which sucrose solution is the same as water potential of the cell. So for that, the graph that you need to plot, okay, I'm going to use the space here to do that, okay? The graph that you're going to need to plot will be the same as this graph here. The only difference is that we are changing this. Okay, we are not using this. We are still using this one. Okay, this one is percentage plasmolysed cells. So you prepare your onion epidermis, you peel it out, you cut it into 1 cm times 1 cm and place it onto the slide. So this is your slide, right? And then you got this 1 cm times 1 cm of your onion epidermis. And then you add maybe 1 or 2 drops of the sucrose solution. So you got 7 here, right? So you add 1 of them there. And then you cover it with a cover slip. Okay, cover it with a cover slip and use tissue paper to wipe the surface so that it's totally dry. Okay, because this is sucrose, you don't want it to touch your microscope and then the end starts to come and then the fungus starts to grow. So the slide must be dry. Then only you can put onto microscope. Okay, so then you look it under the microscope and look at all the cells. So let's say, for example, in a field of view, right? This is a field of view. It means when you look into microscope, this is what you can see, right? So the field of view, there'll be a lot of cells in it. Okay, so what you need to do is that you need to look at all the cells that you have here, or in exam time, the students are asked to choose 10 cells randomly. Okay, so maybe in this magnification, you look at the cells, and then you see a lot of plant cells, okay? A lot of plant cells, epidermal cells. So then you're asked to choose 10 cells, and from the 10 cells, you note down, out of 10 cells, how many cells are plasmolysed? You can see, right, plasmolysis. If plasmolysed means you can see membrane withdrawn away from the cell wall, right? either it's a small degree or huge degree, doesn't matter. Okay, so this one is plasmolized, right? And then the rest are not plasmolized. So this is 1 out of 10 lah. Okay, so therefore you calculate the percentage. 1 out of 10 is 10%. So you plot 10% here. So you use different concentration. You go to the slide and then you count number of cells out of 10 cells that are plasmolized. Okay, and then you plot the graph. When you plot the graph, it will look exactly the same as this graph here. It look exactly the same. Okay, exactly no. Similar, I can say similar. Okay, similar. Then from there, you will be able to find out what is the water potential of the cell by checking when there's, now this is percentage, right? Percentage means this must be 100%. Okay. And therefore half of it is 50%. So let's say this is 50% here. Okay, 50%. So which concentration will give a 50% cell that's plasmolized? Okay, this is the concentration that tally with the water potential of the cell. Okay, whichever concentration that gives 50% out of total cell that you see that's plasmolized, this is the concentration that is similar water potential as the cell. So from there, you can go to a table to convert this 0.5 molar to kilopascal. Okay, so this is another way to find out the water potential cell. So this is the last one I'm going to teach you today, and that's all.